Once there lived a happy little Abuela. She lives her days doing kind deeds, helping everyone and everything around her as much as she is able to. She impacts her community with her positivity and kindness. She becomes the beloved of the town. But as time goes by, things started to look a little fuzzy for her.
Abuela, did you like my performance?
Wait, I thought it hasn't started yet.
NANI?!
This is not only Abuela's life story. The same narratives happen in hundreds and even thousands of households. This could even happen to those closest to your heart. But do not lose hope. We have something that will surely return your courage. Stem cell therapy is your answer. So, what is a stem cell? Our body contains billions of cells that makes us what we are. These cells are specialized to perform a specific function in our body. They are known as specialized cells. However, our hero comes from another type of cell. Here, we're talking about stem cells. They have the ability to differentiate themselves and self-renew. Because of these different functions, they have a remarkable range in terms of understanding as well as treating diseases. It has given scientists much excitement from its promising prospects. So, where did all this start? In 1961, stem cells are first utilized upon the imposition of radiation towards mice. Scientists Till and McCullough decided to infuse bone marrow from a donor to save the mice. Stem cells that are derived from the bone marrow within adult cells can be called as multipotent stem cells. This cell type is beneficial to therapy as it will not result in a tissue rejection upon transplantation. ACS, or adult stem cells, also does not cause many major issues upon isolation due to its means of access. But the presence of this cell type is low in tissues that contains them, and finally, it does not retain the ability to differentiate into a plethora of cells as it is limited to similar types of cells from where it was sourced. Afterwards, experiments to clone different organisms started to be conducted. From the cloning of frogs by Jorn Gurdon in 1962 to the preeminent cloning of Dolly the sheep by Ian Wilmot, something in the midst of all the astonishing feats, embryonic stem cells were identified in mice. Let's look at 1981, where Martin Evans and Matthew Kaufman decided to culture early embryonic cells or embryoblasts. They conducted genetic modifications within the mice to create offspring that fulfilled the gene changes. Then, this sort of experiment reached a new breakthrough when James Thompson of the University of Wisconsin in Madison and John Gearhart of Johns Hopkins University in Baltimore isolated the first human embryonic stem cells. Embryonic stem cells, or ESCs, are stem cells derived from the undifferentiated inner mast cells of a human embryo. This type of stem cell could in one matter be regarded as the perfect cell type for therapy because of a very important characteristic. Embryonic stem cells are pluripotent, meaning they are able to grow and differentiate into all derivatives of the three primary germ layers, which are the ectoderm, the endoderm, and the mesoderm. Unfortunately, it is controversial for some parties due to the fact that it can develop into a living, breathing person. There are also studies that state that it may develop into a tumorigenic. Unfortunately for the scientific community, the aforementioned types of stem cells have limitations that does not allow the scientists to provide therapies that fulfill maximal expectations. However, in 2006, an approach that has not yet been done occurred that may just revolutionize our understanding and application of stem cells as we know it. Shinya Yamanaka of Kyoto University in Japan reveals a way of making embryonic-like cells from adult cells, and therefore this overcomes the issue of an inherent need to destroy an embryo. His team reprograms ordinary adult cells by inserting genes to form induced pluripotent stem cells or iPSCs. Currently, this type of stem cell is still the best candidate to be used in stem cell therapy, since it poses no ethical problems and also has a great ability to proliferate without feeder stem cells. Also, any links to tumorigenic issues were not found. So, how would a scientist produce an iPSC? For instance, in a petri dish with adult differentiated cells within it, giving specific genetic instructions will transform the cells into stem cells that are unlike any stem cells already present in the body. They have the same potential and ability such that of an embryonic cell would have without the conspiracy. Additionally, because the cell is taken from a person, it means that after isolation and modification where the cells insert back into the same person, there will be a very low possibility of rejection by the host body. Therefore, this will open new doors and opportunities for personalized medicine.
So, why is this important? With growing age, vision may degenerate and may eventually lead to blindness. But not to worry, iPSCs can be cultured into RPEs or retinal pigment epithelium cells. The RPEs are implanted into the faulty eye in order to replace lost photoreceptors within the eye. Thankfully, people who suffer from age-related damage, macular degeneration or ADM, the retinal cells are not completely deceased. However, they are dying and substantially require prevention of retinal death and replacement of new cells. After execution of these experiments for 12 months, microscopic examination confirmed the greatest success. The implants are able to survive, and what's more, the patient's vision improved considerably.